In this video, I'm going to be talking about how we use Punnett squares in genetics. Because this is the first video on Punnett squares, we're going to only be talking about monohybrid crosses, or looking just at one gene. Remember, mono means one. And the alleles of the genes, their relationship is going to be one of simple dominance. Before we get into the specifics on Punnett squares, I want to do a quick review. Remember, genes produce specific traits in an organism, and the traits are things that we can see, like flower color down here, a purple or a white flower. These variations, which particular color it is, are caused by different alleles of the genes, a slight difference in how the gene is put together on the DNA level. The gametes, which are the sex cells, carry only one allele for a trait, but then an organism will inherit two alleles, and the way those two alleles interact is what determines how that trait is expressed in the organism. I also want to do a quick vocabulary review before we jump into the actual problems. Remember, a genotype is the specific alleles present in the organism. What is the gene present? The phenotype is what we actually can see, the visible expression of that trait. The relationship between alleles when it is one of simple dominance, or it's also called complete dominance, is that the alleles have a dominant and recessive relationship. One allele will dominate over the other if it is present, and it determines what is being expressed, whereas a recessive allele has to have two copies in order for that particular trait to be seen. And then we have the words homozygous, where the alleles are the same, and heterozygous, where the two alleles in the organism are different. So a Punnett square is a tool used to predict the genotypes produced by a particular genetic cross or breeding experiment. And it just looks like this chart over here. This is a Punnett square where we're only dealing with one particular gene, or two alleles per parent. If you know the parent genotypes, then you can predict the offspring genotypes. And this is the most common way that Punnett squares are used. But you can also work the other way. If you know the offspring genotypes, you can go backwards and figure out what the parent genotypes had to be in order to produce that particular offspring combination. Punnett squares are most useful when you're only looking at one or two genes, because it just gets to be too big of a chart if you're dealing with more alleles than that. So let's put this to work in a particular problem. So our problem says that in guinea pigs, the allele for short hair is dominant over the allele for long hair. So we've got the trait is hair length, short hair and long hair, and short hair, the allele is dominant. So if a long-haired guinea pig is crossed with a homozygous short-haired guinea pig, what are the potential genotypes and then phenotypes of the offspring? So we have our long-haired guinea pig being crossed with a short-haired guinea pig. So the first thing you need to do in order to figure out this problem is to get the parental genotypes. So the information we know from the problem is that the short hair is a dominant allele, and we have a long-haired parent and a homozygous short-haired parent. So our alleles, I'm going to use the letter S for short hair. So we'll use a capital S as equaling to short-haired allele, and then a small s, and I'm just going to underline that to make it more clear that it is a lowercase letter, is going to represent our long hair allele. So since the long hair allele is recessive, this long-haired guinea pig parent has to be just two small s's. That has to be the genotype of that parent. That is the only way that recessive trait would be expressed. And we know that it's a homozygous short-haired guinea pig, so this one also will have two alleles that are the same. In this case, they're both capital S's. So after you've determined your parental genotype, the next thing to do is to put them over into the Punnett square chart. Each genotype will take up a certain place on the chart. It'll either be in the columns or it'll be in the rows. So I'm going to do the columns first, and we'll start with our long-haired guinea pig. So each one of these alleles gets its own column. So we have two small s's across the top, and then I'm just going to switch colors here. The short-haired guinea pig will go down the rows. So we're going to have two capital S's going down the rows. Now the next step is sort of like Battleship, where you're going in from the top and the side and seeing where you end up. So this S will be going across and filling in both of those squares on your row. The capital S in the bottom will also be going and filling in. And then we do the same thing with the letter from the column. So we're going to go down with our long-haired allele in both columns. And so what is left is our genotype of our offspring, and it's all exactly the same. We can say this is 100% heterozygous. So it has both a capital S, I can't talk and spell at the same time, both a capital S and a small s. It's 100%. We predict that all of the offspring are going to have this particular genotype, which is going to mean they are 100% short-haired, because this short hair is a dominant allele, and so as long as you have one present, one capital S present, then the way it's going to be expressed will be a short-haired guinea pig. Let's look at another cross. Again, we're still talking about short hair and long-haired. So if a long-haired guinea pig is crossed with a heterozygous short-haired guinea pig, so perhaps one of the offspring from that previous genetic cross, what genotypes and phenotypes do you get as a result? So again, using those same alleles, we know that our long-haired is a recessive trait, so there's going to be 2 lowercase or small s's. But we have a heterozygous short-haired guinea pig as the other parent, which means you have 1 capital S and 1 small s. So again, taking these over to fill them in, I take our long-haired guinea pig across the columns like we did on the other side, and then switching colors for our short-haired guinea pig. Going down the rows, it will be a capital S in this first row, but a small s in the second row. And then just to fill them in, again, just putting, going across the rows, just using that same allele and filling in the chart, and then going back to fill in from the columns, going down, we have small s. And so I hope you can see right away we're getting some differences here. Again, we're seeing this heterozygous genotype of 1 big S, 1 small s, 1 capital, 1 lowercase. But we also have the recessive homozygous genotype with 2 small s's. So the genotypes that we see here is 50% heterozygous and 50% homozygous recessive. So you have to say what kind of homozygous genotype it is, because it can be either the dominant or the recessive allele. So here we have a 50% homozygous recessive. 50% because out of these 4 boxes, 2 of them are of each kind. So we have 2 heterozygous and we have 2 of the homozygous recessive. What would be this is the genotype. What would be the phenotype? Well, we've got 50% of those heterozygous will be showing short hair, and the other 50% will be long hair. Now, again, these are statistical predictions. In real life, if you were really breeding guinea pigs, you would not necessarily get a 50-50 split from these 2 parents. Just, a 50% chance whether you get a boy or girl in a human family, but it doesn't work out that way always. When parents have children, it doesn't divide so nicely. So in real life, you have to have lots and lots of offspring to get close to these predicted percentages. But the Punnett Square allows you to make predictions on what you could, in general, expect to find if you had enough offspring to work from. So in summary, Punnett squares are a tool to predict the probability of certain genotypes and then the phenotypes that rise from them. You take the alleles from one parent and arrange them across the top of the columns, and the alleles from the other parent are arranged down the rows, and then you combine the alleles in the column and the rows to create these potential genotypes. Use the genotype to determine the phenotype and make your predictions about what will result from that particular genetic cross. You'll have lots of time to practice this in class working with your own Punnett square problems.